Hey guys, welcome to AP Biology. Today we're going to talk a little bit about feedback loops, thermoregulation, and feedback inhibition. So first of all, we need to know a few important terms. The term homeostasis, you all know that, meaning trying to keep various body levels, whether it's like glucose levels, calcium levels, temperature, things like that, within a relatively narrow range that is conducive to life. A negative feedback loop, again, as I say up here, a negative feedback loop occurs in biology when the product of the reaction leads to a decrease in that reaction. Use your home furnace as an analogy. At home, we're going to set our furnace, let's say 75 degrees, and when the temperature drops below 75 degrees, then the furnace turns on. The product is heat, and when that heat brings the temperature back up to 75 degrees, it turns the furnace off. That would be negative feedback. Positive feedback is a little bit opposite. And again, that's when the product of the reaction leads to an increase in that reaction. So using our furnace analogy, that means that as we hit 75 degrees, that would tell the furnace, hey, we need to get hotter, and then hotter and hotter and hotter and hotter until we would be sitting in a sauna. Now, there are not too many positive feedback loops in mammal biology, but there are a few that we'll talk about. So what are some of the components of a typical feedback loop? In this case, we'll look at a negative feedback loop. First, there has to be a stimulus, something that changes the level away from the set point. There has to be something that receives or senses that change, a receptor, a control center, which then, as I say, compares the changes to the set point range and sends out nervous or endocrine signals to effectors, whether that's a gland or it's a muscle or something of that nature, which will hopefully reverse that change back towards that set point. And again, our control center is oftentimes our hypothalamus for many of our negative feedback loops. The effector, what's making that adjustment, okay, and then the response. And oftentimes the effector is going to be an organ, it's going to be a chemical or something of that nature. And of course, the response will be hopefully getting us back to where we need to be. So here's a model of a typical negative feedback loop. And pause the video whenever you need to stop and look at some of these diagrams. So our variable down here in the bottom on the seesaw, it's in balance. It's in homeostasis. Now, if we see an imbalance occur due to some sort of stimulus, we see that variable is now imbalanced. That change is going to be detected by a receptor of some sort. And that receptor is going to send a signal to the control center. The control center is now going to send another signal to an effector, and then the effector is going to basically release a chemical, cause something to happen that then brings our imbalance back into balance. And that's a basic model of a negative feedback loop. And for thermoregulation, the way that might work is that we have our set temperature somewhere between 35.6 and 37.8 degrees centigrade. And again, all of us don't have the same temperature. I have a tendency to run a little cool. My normal temperature is around 97 degrees Fahrenheit, which is probably about 36 degrees, 36, somewhere around there, centigrade. But let's look at this. So we get too hot. All right, so the stimulus is increased body temperature. The sensor is going to be in the hypothalamus, and that's the set point, and that's also the control center because blood's flowing through there. It's going to send a signal to the effector organs, and those effector organs are going to be your blood vessels in near your skin, the capillary beds. That's going to cause skin dilation. That means that the vessels are going to get bigger in diameter. They're going to increase blood flow to the surface of your skin, and that's when you would flush. You get red in the face or paler in color or something of that nature. And also, it's also going to stimulate your sweat glands to produce sweat. And remember, the high heat of vaporization of water takes a lot of energy to convert water into a gas. And that energy in the form of heat means it's going to take heat away from your body, with the overall result of lots of blood flow going to your skin around these sweat glands, bringing that heat towards the surface, and then that's hopefully going to drop your temperature back to that normal. And think of all the adaptations that many different animals have to either get rid of waste body heat, think of the large ears in elephants or the very large ears in like a desert jackrabbit. Or if you notice on your dog or on a horse or something, they have very little fur on their belly. That's called a thermal window, and they can orient that to certain directions. Lay flat on your dog. You may see him laying flat on cool concrete in the summertime when it's hot outside, trying to get that heat away from its body using those thermal windows. Horses orient themselves, their bodies towards the wind, so the wind is going over those bare spots, helping to dissipate some of that heat. Okay. Now, of course, if you get too cold, the hypothalamus detects that once again. It's going to send a signal to your capillaries to constrict, get much smaller, reduce blood flow to the surface, and then also to induce shivering, both of which will help to hopefully increase your body temperature. And I was telling my other students that I used to work up in Alaska with walruses, and the walruses go out and feed in the Bering Sea, which has a temperature of about 37 degrees Fahrenheit, so it's very cold, just above freezing. And these animals would feed for about 10 days before they would come back into the shoreline to sort of sit around and digest it, literally. And when they came in from the water, they were almost white because of the vasoconstriction, preventing all the blood flow from going to their skin. And then after about an hour on shore, they would turn this deep red, bright pink color as they started to heat up. And then in order to reduce the heat, sending all that blood to their skin, causing them to really blush, if you will. And in the pivot lab that we did, we talked a lot about thermoregulation. You may not realize it, but in the pivot lab, we were looking at ectotherms and endotherms, and we looked at basal metabolic rates. And we saw that in the ectotherms, they were much more affected by temperature than were the endotherms. Okay, so ectotherms, otherwise known as cold-blooded or thermoconformers, because they conform to the temperature outside their bodies. Of course, when the temperature drops, their body temperature drops, which leads to a decrease in their metabolic activity, which we looked at and we measured in the pivot lab by looking at the amount of CO2 being produced. At cold temperatures, the ectotherms produce very little CO2, but as we increased that external temperature, we saw that the amount of CO2 being produced increased, and that was a measure of metabolic activity. And again, what are examples of ectotherms? Plants are ectotherms. Most fish, reptiles, amphibians, and insects are all ectotherms. Now, there are some interesting exceptions to that, like tuna can actually increase their body temperature. And there are some interesting reptiles that can do some stuff too, like alligators and crocodiles. So what are the advantages of being an ectotherm? Number one, you don't have to spend energy to heat your body. And therefore, most of the energy you ingest allows you to put that towards cell growth, reproduction, things of that nature. And also, it's going to reduce the amount of time you spend looking for food. And when you reduce the amount of time of looking for food, you're reducing the amount of time you expose yourself to predators. Think about it that way. What's the disadvantage of being an ectotherm? Number one, you kind of narrow the choice of habitats you can live in. Endotherms, inside heat, these were the mice and the rats that we saw in that pivot lab. They are thermoregulators, and generally changes in external temperatures have little to no effect on the endotherm. What's the advantage? Basically, that allows endotherms to live in many different environments around the planet. The disadvantage, they have to use a lot of energy, therefore they have to eat on a regular basis, and they also have to expose themselves to predators. And even if you are a predator, going after prey is not risk-free. Okay, many predators die in the pursuit of their prey items. Okay, so when we compare these two, again, as we saw in the pivot lab, the endotherms have much higher metabolic rates than the ectotherms overall. And we saw that when the external temperature drops, the metabolic rate of the ectotherms decreased. And we saw a slight increase in endotherms, but it really wasn't all that much compared to what their normal metabolic activity was. Same thing with the opposite. When the external temperature increases, metabolic rate of the ectotherm will rise, and there will be little to no change in the endotherm. And also do remember, as we saw in the pivot lab, mass determines metabolic rate. Okay, we saw in the mouse had a much higher metabolic rate than did the rat. And a lot of that has to do with the surface area to volume ratio. Small organisms have a high surface area to volume ratio. Therefore, they're losing a lot of heat to the environment, or they're gaining a lot of heat from the environment. Okay, and I want you to look at this graph very carefully. Look at the x-axis. The x-axis is a logarithmic scale. We go from 0 to 0.01 kilograms. Okay, that is 1 gram. Okay, that's very small. And then we go from 1 gram to 10 grams. And then we go from 10 grams to 1 kilogram, which is about 2.5 pounds. Okay, then we go from 1 kilogram to 10 kilograms, 10 kilograms to 100, and 100 to 1,000, and 1,000 to 10,000. That is a logarithmic scale. Okay, the AP likes to throw these on there once in a while, and many students have never seen one before. Well, now you've seen one. What does a logarithmic scale allow us to do? It allows us to plot organisms or data points that are really far apart on the same graph. And we did this in the pivot lab. We created a logarithmic curve to that fit, and that's what we see here. Small animals very small animals have very high basal metabolic rates. Large animals have very low metabolic rates. Okay, the shrew, heart might be 300 to 400 times per minute, whereas in the elephant, it may be, 15 to 20 times per minute. A blue whale would be even lower than that. Blue whale has a heart so big you could walk through it. So let's look at negative feedback loops. This is blood glucose maintenance. This is one you must know. And whenever we're talking about sort of these hormone-induced feedback loops, there's always there's always two hormones. One hormone, in this case insulin, which will reduce blood glucose levels in this particular case, and the other hormone, glucagon, which will increase blood glucose levels. They have opposite effects. They are antagonistic to each other. So once again, our blood glucose levels, you eat a meal, your blood glucose levels are going to go up. Cells in the pancreas and in your hypothalamus will check that will sense that increase in blood glucose, then signals will be sent to specialized cells, beta cells in the pancreas, which then release insulin into the bloodstream. The insulin will travel to muscle cells and to the liver. And when they get there, a whole signal transduction cascade occurs, which is going to cause little vesicles filled with glucose transporters to merge with the cell membrane. When those glucose transporters become incorporated into the cell membrane, they can now take glucose from the blood and bring it into the cell. And we store that in the muscle cells or in the liver cells. And in the muscle cells, they can be stored as either free glucose or as glycogen, which is a polysaccharide of glucose. In the liver, they will be stored as glycogen. What's the effect of that? Blood glucose levels decline. So a couple hours after you've eaten, your blood glucose levels drop. Your pancreas cells and cells in the hypothalamus are going to detect that. They're going to send a signal to the alpha cells of the pancreas to release a different hormone, glucagon. Glucagon travels via the blood to the liver, and in the liver, the glucagon is going to activate enzymes that are going to convert glycogen, a polysaccharide, into glucose, a monosaccharide, and then that gets moved into the blood and causing blood glucose levels to rise. The next one I always tell my students, you need to know a couple of these different feedback loops just as examples of how feedback loops work. And in this particular one, what do we see? This is for calcium. Now, calcium is a really important molecule in your body, ion in your body. It plays many roles. It plays a role in your nervous system and at the synapses. It plays a role in muscle contraction. It also plays a role as a secondary messenger in many different cell signal pathways. So it's really critical that calcium levels are maintained at a pretty precise level in your body. So the main storage area for calcium is in your bones. And so once a increase in calcium levels are detected by the thyroid gland and in the hypothalamus, there's receptors in the thyroid and control center in the hypothalamus. So the hypothalamus sends a signal to the thyroid to release calcitonin. Calcitonin is a small protein molecule that's going to stimulate the deposit of calcium salts into the bones, and that leads to a decrease of calcium levels in your blood. If your calcium levels decrease, a different gland, the parathyroids, which are very small little glands that rest on the backside of your thyroid gland, they are signaled to release parathyroid hormone, and parathyroid hormone travels via the blood to osteoclasts, which are a type of bone cell, and that causes them to release calcium into the blood, bringing your calcium levels back on up. Okay, so again, we're seeing these negative feedback systems happening in this particular case. Calcium being the molecule that has to be regulated. The production of the hormones leads to a decrease in those levels. Production of PTH leads to a decrease or leads to an increase in calcium levels on the other end of that side. Okay, positive feedback loops. Breastfeeding is one. And again, this would be the example like in the furnace where it keeps on getting hotter and hotter and hotter. In this particular case, the baby will suckle at the breast of all mammals. That is the stimulus. Sensory receptors in the skin and breast detect that, send impulses to the hypothalamus. Hypothalamus signals the posterior pituitary gland to release oxytocin. Oxytocin travels via the blood to the cells in the breast, which are going to stimulate the ejection of milk. Milk is released. The baby gets the milk. The baby's go, yum, yum, starts to suckle some more, and that keeps on going and going and going, releasing more and more milk. And as long as the baby suckles, not continuously, but on a daily basis, the female will continue to produce milk. Once the baby stops suckling, no more oxytocin is being released, no prolactin is being released, and milk production will cease. We see the same thing in childbirth. I think I mentioned this to you guys earlier, but just in case I didn't, oxytocin also plays a role in childbirth. Again, the baby's head, baby's head, I guess at this point, is pressing against the cervix. Receptors, sensory cells in the cervix are going to detect that pressure, send a message to the hypothalamus, and the hypothalamus is now going to send a message to the posterior pituitary to release oxytocin. Oxytocin stimulates the uterus to contract, okay? And that is, and more pressure on the head from the baby pushing is going to cause the uterus to contract, putting more pressure on the cervix, sending those signals back again and causing more contractions until that baby is born. Now, I do want you to see that estrogen from the ovaries induces oxytocin receptors on the uterus. So estrogen is at high levels during pregnancy, and so there's going to be lots of oxytocin receptors on the uterus. What happens after the baby is born? The amount of estrogen goes down. The receptors on the uterus begin to disappear, which is important because remember, breastfeeding is also under the control of oxytocin. So if that didn't happen every time the baby was suckling and oxytocin would be released, the mother's placenta would be cramping. And that would be very unpleasant and uncomfortable, and the mother would not want the baby to breastfeed, and so on and so on. So biologically and evolutionarily, it makes a heck of a lot of sense if those oxytocin receptors on the uterus disappear after childbirth. Okay. An example in plants, positive feedback loop is fruit ripening. An apple begins to ripen on a tree. It produces ethylene gas. Ethylene gas travels via the air to nearby neighboring apples, causing them or inducing them to begin the ripening process, which induces their neighbors, and so on and so on. And all of a sudden, all the apples on a tree are ripe very quickly. And ripening involves a lot of different pathways and a lot of different hormones. Apples go from acid to neutral, sour to kind of sweet, under the influence of kinases, enzymes that are changing that. They go from starchy, think of a green banana, doesn't taste like crap, okay, versus a ripe banana, which is nice and sweet. That's because the enzyme amylase is breaking starch into glucose and sucrose units. They go from green to red because hydrolase breaks down chlorophyll, and therefore less chlorophyll. We start to see the accessory pigments. Pectin, which makes things hard. If you ever made jelly, you added pectin to it. The enzyme pectinase is going to break that down, produce less pectin. It's going to get softer. Large molecules by hydrolases are going to be broken down into smaller molecules, and many of those molecules play a role in fragrance. Okay. Last thing I want to talk about are metabolic pathways. We've already talked about these before, but just as a reiteration, again, they are multi-step enzyme-mediated biochemical reactions that lead to biosynthesis, anabolic reactions, or breakdown, degradation, catabolic reactions of biological molecules within the cell. We've already talked about glycolysis, the Krebs cycles, and Calvin cycle. Photosynthesis, cellular respiration, and digestion are all examples of metabolic pathways. They are all consist of multiple separate chemical reactions where the products of one reaction are typically the reactants or substrates for the next reaction. And all of these reactions, remember, are catalyzed by specific enzymes. And so in order to shut down one of these systems, generally we got to turn off the enzyme. And we do this via allosteric inhibition. So here's a biochemical pathway, metabolic pathway, where we're taking the initial substrate, threonine, which is an amino acid, and through a series of 5 different reactions, converting it into isoleucine, another amino acid. The product is going to act as an allosteric inhibitor of the first enzyme, because as long as there's enough of this product, why make more? Okay, and so that enzyme gets shut down, and we shut down that pathway. Again, negative feedback. The product shuts down the pathway. And once we start using up all the isoleucine, then this will be used up, the one that's on the allosteric site, and now the active site goes back into its normal shape, and now we can start that pathway all over again. And it's just another view of the same thing happening where the end product is shutting down that enzyme. And that's it, guys. I hope you learned something, and good luck.